The overall goal of the following experiment is to produce pH-responsive nanosensors and self-reporting scaffolds that can be monitored in real time and in situ in order to assist with the understanding of conditions that influence cellular growth. This is achieved by first preparing biocompatible analyte-responsive nanosensors, which can monitor pH through fluorescence outputs. As a second step, pH-responsive nanosensors are incorporated into polymeric electrospun scaffolds, forming a 3D environment upon which cells can be cultured and monitored. Next, mammalian cells are seeded on the electrospun scaffolds, and nanosensors are also delivered into their intracellular environment in order to monitor intracellular pH during experimentation. Results are obtained that show the ability of intracellular nanosensors and self-reporting scaffolds to monitor pH within tissue-engineered environments based on fluorescence ratios acquired by confocal microscopy.
The main advantage of this technique over existing methods, such as the use of electronic pH probes, is that measurements can be performed in situ and in real time without having to disturb cellular growth.
To begin, prepare the fluorophores by dissolving 1.5 milligrams of FAM-SE into 1 milliliter of dimethylformamide, known as DMF, in a round-bottom flask. Also, dissolve 1.5 milligrams of TAMRA-SE into 1 milliliter of DMF in a second flask. Then,